lecture, we will begin our discussion on Unit 3, Cellular Energetics. In cellular energetics, we're going to begin our discussion with what we call enzymes. So we'll take a look at enzyme structure, how they work as catalysts, and also look at how they are affected by the environment around them. Later in this unit, we'll take a closer look at photosynthesis and cell respiration. In this lecture, we will be covering or at least a review of energy and chemical reactions. We'll take a look at enzymes and, as I mentioned, how they function as catalysts and how their structure allows them to do that. And then we'll take a look at the environmental impacts on their function. In this opening slide here, you see this individual considering a purchase of some vitamins. What are the significance of vitamins? Why do people take them? As well as certain medications. Well, a lot of that has to do with enzyme function. So as a human, as a metabolically active organism on this planet, you depend quite a bit on the chemical reactions going on in your body. And enzymes play a big role in that. And at the end of the lecture, we'll take a look at why vitamins can assist with that. To begin, we should probably review what a chemical reaction is. So it is a process in which one or more substances are changed chemically into something else. And this involves the breaking of bonds, typically covalent bonds. And that change requires energy. Energy needs to be used. Now, inside of your body, you have many, many chemical reactions going on. So the sum total of all of those chemical reactions occurring in an organism is known as its metabolism. And it's hugely important in maintaining homeostasis and keeping things alive. As I mentioned, energy is crucial to this. And so energy, you may recall from physics or physical science, that it is the ability to do work or cause change. And so if we're going to change chemicals into other chemicals in the body, then energy is going to be used to do that. So energy is hugely important for life. And so as we take a look at chemical reactions, and as we understand that there's potential in kinetic energy, we need to understand there's what we call chemical potential energy. And this is the potential for a chemical reaction to occur. And so even at the chemical level, thermodynamics must be obeyed. So the first law of thermodynamics says that energy cannot be created or destroyed. So certain reactions may not proceed if there's not enough energy put into the system. The second law of thermodynamics also holds true when it comes to chemical reactions, in that whenever there is an energy transfer, there is an increase in entropy or an increase in disorder. Well, what does this mean for chemical reactions in the body and energy in living systems? Well, a little bit of chemistry review here. And I want to be clear that these terms are not necessarily going to be on the test or the biology AP Bio exam. But it's good to review them and kind of understand and build towards our understanding of how enzymes function in the body. So the term enthalpy refers to the total energy in a system. So again, apply this to living things, right? We're looking at the total amount of energy in an organism, in food that we eat, for example. Well, free energy is the amount of a system's energy that's available and being used for work. Okay, so we've got enthalpy is the total energy, that's H in this equation, right? And we're going to look at free energy is known as G. It's usually given the term G. I shouldn't say usually, I believe it is given G because it's usually referred to Gibbs free energy, who is the person that described this. Entropy is designated with the letter S. And Entropy is the amount of usable energy in the system. So we have these three variables, and if we throw temperature in here, we have these three equations that can help us to understand how chemical reactions proceed. So in this equation, in this variation, we have the enthalpy, the total energy of the system, is going to be equal to the free energy that's available to do work, plus the temperature of the system times its entropy, or the amount of unusable energy in the system. If we rework the equations algebraically, we can see that the free energy, which is very significant in a system, the free energy, the amount of energy available to do work for living things, is equal to the total energy available minus the temperature times the entropy, right? So, and second law of thermodynamics says that entropy increases over time. And so this is significant when we look at chemical reactions. This equation here just shows that the change, or delta G, which is the change in the free energy, is equal to the change in the total energy of the system minus the temperature times the change in entropy. So really that's looking over time, which will be important to understand because chemical reactions proceed over time. What do these equations and this delta G, what's going on here? Well, there's two general types of reactions. There's what we call exergonic, which are chemical reactions that release free energy and tend to occur spontaneously. So, these are occurring spontaneously. Notice their delta G is less than 0. So they have a negative delta G. Exergonic reactions can also be referred to exothermic reactions if they release heat energy. Endergonic reactions, also known as endothermic reactions, are reactions that require the addition of free energy and don't proceed spontaneously. They have a delta G that is greater than 0, or a positive delta G. What would this look like graphically or visually? Well, let's take a look at our the graphs here. We're going to compare an endothermic or an endergonic reaction to an exothermic or exergonic reaction. So let's look at endothermic, right? In this graph, we can see that our vertical axis is the energy, right? The energy available in the system. And the horizontal x-axis is the direction of the reaction, or you could also say time, the time of the reaction. Well, in the beginning, before the chemical reaction occurs, there are energy in what we call the reactants. Remember, reactants are those things that are going to react in the chemical equation. So there's a certain amount of energy present in the reactants before they go through their change. Now, for the chemical reaction to occur, we must reach something known as activation energy. And so I like these graphs because it kind of shows like, okay, we need to add energy into the system for it to proceed. So this is kind of like if you wanted to roll a ball up a hill, you're going to have to put energy. So if you're going to roll a ball up this hill here, you're going to have to exert a lot of energy. But notice once you get to the top of the hill, you can let it go and it'll roll down that hill at a certain speed. Now notice in this graph, in an endothermic reaction or an endergonic reaction, the energy in the products, and remember the products are the end result of a chemical reaction. So the reactants turn into products. Notice the products here have absorbed energy and their total energy, or this is basically gives you a positive delta G, right? So that's known as an endothermic reaction. So they have absorbed energy. Now, an exothermic reaction has the same characteristics. You still got to roll the ball up the hill, but once you hit that activation energy, the reaction proceeds very quickly in essence spontaneously. And so energy is released from these reactions. So exothermic reactions are usually said to release heat. So if a reaction becomes hot, it's exothermic energy, heat energy is being released. The products aren't keeping that heat. They're not absorbing it. They are releasing it. So if a reaction becomes cold, we say that's endothermic because it's absorbing heat and feels cold to us. If it's releasing heat, then it's exothermic. Now, if it's not heat energy that's being released, like for example, some reactions release light, such as like the chemical reactions that produce the light in a firefly. That's releasing light energy, not heat energy. And so we don't say that's exothermic, we would say that's exergonic instead. So notice those terms are interchangeable. Now, where does this come into keeping you alive? Well, you may recall that we've been talking about adenosine triphosphate in previous lectures and lessons, also known as ATP. ATP is known as the energy currency of the cell. You may recall from last unit that the mitochondria is responsible for making ATP. It's the powerhouse of the cell. So why is the mitochondria so important? Why is ATP so important? Well, it's because you'll notice that in adenosine triphosphate, the term triphosphate, there's 3 phosphate groups, right? So 3 functional phosphate groups here. Well, in this third phosphate, there's a lot of energy potential there. If we could somehow break that off, we would release a lot of energy that could be used to do cellular work or work inside the cell. Well, you may recall what kind of reaction breaks down macromolecules into smaller things. If you said hydrolysis or breaking with water, you're correct, right? So water is used in the system. Another reason why water is very important to living things. That hydrolysis reaction proceeds. Notice we have kicked off a phosphate. It releases energy. And take a look at the delta G, right? We have released a lot of free energy here. It's a negative delta G. And that's wonderful. This is what allows your muscles to move and why you're able to produce action potentials in your nerves. So ATP is hugely important. But the amazing thing about ATP is that it can be built back from adenosine diphosphate. Again, it's diphosphate because it only has 2 phosphates left. We can actually add that phosphate back on. And so notice in this diagram over here, if we put energy into the system, we can synthesize more ATP, right? But notice that's an endergonic or endothermic reaction, meaning we need to add energy to recharge ADP into ATP. Well, how do you do that? This is why you eat food. So when you eat, you're putting in chemical energy into your body digests that, and it basically recharges your ATP. And that's what your mitochondria is doing inside each and every one of your cells. It is taking the energy you put into your body and recharging your ATP molecules. And therefore you have a currency of ATP molecules that are ready for hydrolysis or breakdown to release energy that could be used for cellular processes. Whatever the cells might need to do, maybe making proteins for your body so that you can release hormones or neurotransmitters, or move your muscles so that you can throw a football. So all these chemical reactions come back and help us understand the significance of energy to life. And so this is the general process. All right, now where do enzymes come into this? Well, an enzyme is a biological catalyst. It can speed up chemical reactions without being used in the reaction. So they're wonderful. They're basically proteins that assist with your chemical reactions. Because you may recall that some of your reactions would proceed too slow. They're not spontaneous. And if reactions are too slow, then you may not have the energy and structures available to live. So hugely important. So the enzyme is a protein that can speed up a chemical reaction. In a chemical reaction formula, if you were writing it out, like this is looking at the breakdown of the toxin hydrogen peroxide, which you may recall your peroxisomes do. So it breaks down hydrogen peroxide into water and oxygen. Now that doesn't happen spontaneously. Like if you pour some hydrogen peroxide into a little clear cup, it will just look like hydrogen peroxide. It kind of looks like water, right? Just nothing is happening. But if you put the enzyme catalase in there, well, catalase will break the hydrogen peroxide into water and oxygen. The oxygen is a gas, and this is where it bubbles. So if you've ever poured hydrogen peroxide into a wound, you probably know, oh, it stings because it's got a kind of sterilizes the wound. But you also notice there's a whole bunch of bubbling going on. And that's because your cells have catalase in them that helps to break down hydrogen peroxide because it's toxic to your cells. The reason you're putting hydrogen peroxide in a wound is because it kills the bacteria. Your cells don't want to die, so they neutralize the hydrogen peroxide almost instantaneously. So it kills the bacteria in the wound, and then your cells not denature, but they break down the hydrogen peroxide into water and oxygen. That's where the bubbling comes from. We'll be doing something similar to this in lab. I don't know if we'll work specifically with hydrogen peroxide, but we can kind of see this reaction occur. Now, what does this look like in terms of our reaction graph, right? So you may recall here we're looking at a chemical reaction that has a negative delta G. Okay, so, and this is looking at the breakdown of glucose into carbon dioxide and water, right? This is essential for life. Your body is always breaking down sugar to release energy. Well, notice the red curve is without the enzyme. So it has a very high activation energy. So it takes a lot of energy to break down glucose and release its energy, right? So that kind of is counterintuitive. It's kind of like you need money to make money, kind of. Well, so that's what's going on with the red line. But if we go to the blue line, notice if we add a proper enzyme, it can lower the activation energy. So notice we don't have to roll the ball up the hill as high as we did without the enzyme. And once we get here, we can let it go and the reaction proceeds quicker. So enzymes will speed up chemical reactions. And as a living thing, that's important to you. You want your reactions to be quick. But what do enzymes look like and how is it that they're doing this? Well, we're going to go back to our understanding of proteins. Remember that proteins are huge macromolecules made up of amino acids as their monomer. And we have the primary structure of enzymes, which is basically all the amino acids bound together with peptide bonds. And then secondary structure where they will make like an alpha helix or beta pleated sheet. Then they will fold into more complex structures with their tertiary structure, and quaternary structure is when they can actually we'll go ahead and combine with other proteins to make an even more interesting complex. So proteins have three-dimensional structure. They have a definite shape. And since enzymes are proteins, we see that in their shape and how it relates to their function. So one key feature of an enzyme protein is that it has what's called an active site. This is the location on the enzyme where the chemical reaction is going to take place. So it's a great name to remember, active site. That's the chemically active portion of the enzyme. Now the substrate is the reactant molecules that are going to undergo the change. The enzyme's not going to change. It's just going to facilitate the reaction of substrates into products. The enzyme substrate complex is the structure produced when the substrates bind to the enzyme. Perhaps looking at an example will help you to understand this. We're going to look at the activity of hexokinase, an enzyme that helps to facilitate the breakdown of glucose with the presence of ATP. So let's go ahead and see how this works. So the first step in this diagram here, we can see the substrates here, ATP, adenosine triphosphate, and glucose, will bind to the active site of hexokinase. So they'll come in, you can kind of see in this graphic how there's a place for the ATP to bind to the enzyme and a place for glucose to bind. This will then form the enzyme substrate complex. When this occurs, notice the enzyme undergoes a conformational change, meaning it changes its shape slightly, and it does so to bring the reactants close together. This is sometimes referred to as an induced fit. It strains the chemical bonds and allows them to be closer together. This is a lot like putting a key in a lock in your door. So you've got like the lock, it has a particular shape, but it's not, you can't open it without the key. You put the key into the lock and there's like an induced fit, like the shape of the key will change the shape of the lock such that now you can turn the lock and open your door. And so an enzyme substrate complex is like that. Once the substrates bind to the enzyme, it changes the shape of the enzyme, and that induced fit then promotes the chemical reaction. It occurs quicker. Notice the substrates are converted here. Now we no longer have ATP, we have ADP, which is adenosine diphosphate. Why? Well, because one of the phosphate groups has been knocked off or removed from ATP and given to phosphate. It is now glucose 6-phosphate. So the products ADP and glucose 6-phosphate are released. However, key feature about enzymes. Notice the enzyme can be reused. The enzyme just helps the reaction proceed. It's not changed itself. It can now go ahead and repeat this reaction over and over again. For an enzyme-mediated chemical reaction to occur, the shape and charge of the substrate must be compatible with the enzyme. So enzymes are specific to certain chemical reactions. So one enzyme typically only promotes the reaction of only one chemical reaction, one substrate. So they're specific, just like one key will only open one door or one type of lock. What does this look like in terms of our graph? Remember, we said that the whole function of an enzyme is to reduce the activation energy. And so notice with the hexokinase enzyme, we have lowered the activation energy and the reaction still proceeds as it was. It's still a negative delta G, still an exergonic reaction. So it's basically, nothing has changed as far as the reaction from reactants to products. We have just made it easier. So enzymes are wonderful that way. Here's a video to help you visualize this a little bit better.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
Here's another video that will help you to visualize it in the human digestive system.
An example of how enzymes function in the body is from the enzyme sucrase. Sucrase resides on the surface of the microvilli on the intestinal epithelial mucosal cell surfaces. This animation presents a graphical representation of the way that sucrase catalyzes the hydrolysis of the common disaccharide sucrose, which we know as table sugar, into its component monosaccharides, glucose or blood sugar, and fructose or fruit sugar. Hydrolysis is accomplished because when the sucrose molecule binds to the active site of the enzyme's configuration is changed so that the oxygen bridge between the two monosaccharides is exposed to water molecules in the solvent. This exposure permits a water molecule to actually break the bond, the oxygen bridge, and attach the components of water, an OH to one of the monosaccharides and an H to the oxygen, which is still attached to the second monosaccharide. This effectively cleaves the bond between the two monosaccharides and converts the disaccharide into two separate sugars. Once this is accomplished, the enzyme's configuration is changed back to the original shape. The two monosaccharides float away, and the site becomes available for another sucrose molecule to bind, change the enzyme's configuration, and be hydrolyzed. This action can be repeated many times until the enzyme becomes denatured, is inhibited, or just wears out.
Now let's discuss how different environmental conditions will affect enzyme function. And so, because enzymes are proteins, any change to their molecular structure will have an effect on their ability to do their job. And so just like any protein can denature or break down or lose its shape, denaturation of an enzyme can occur and eliminate its ability to catalyze reactions. Things like environmental temperatures and pH are things that can affect a protein shape or denature it and change its efficiency. Enzyme denaturation is reversible, just like we learned like with protein in your hair, right? You can apply heat to your hair to change its shape or denature the proteins, but eventually they'll go back. So you can straighten wavy hair temporarily with heat, but it'll eventually go back to its shape. Well, let's talk specifically about how heat affects temperature. So and this is true not just of enzymes, but chemical reactions generally go faster at higher temperatures, right? Because you're adding heat to the system, and that speeds up molecular motion. So higher environmental temperatures tend to increase the movement of molecules in a solution, and therefore the frequency of collisions between enzymes and substrates. So warmer temperatures tend to facilitate enzymatic reactions. However, they're generally, especially in living things, there typically is an optimal temperature. So for example, in humans, most of our enzymes work optimally at 37 degrees Celsius, which is the temperature, the human body's core temperature, right? 37 degrees Celsius is equivalent to 98.6 degrees Fahrenheit. Your enzymes are designed to work best at that temperature. Now, if you become too cold, if your body temperature becomes too cold, you can enter into a state called hypothermia. Notice what would happen to your enzymes' functions and activity. If you got too cold, your body temperature got too cold, your enzymes would slow down. And now your chemical reactions aren't occurring fast enough, and you can end up in a dangerous situation. That can affect your life. Well, what if it gets too high? Well, this is why extremely high fevers are a concern in humans, because if your core body temperature gets too high, now the enzymes, which are proteins again, could denature and lose their ability to catalyze reactions at all. And so we will eventually cover why, fevers are important, right? They're your immune system's response. You elevate your core body temperature because you're trying to kill a pathogen or get rid of it. But if it's too severe and your body temperature goes up too high, then your system can work against itself. So that's why, especially in young children, we're very concerned about high fevers, and we do want to control them with medication, Tylenol and ibuprofen and such. Well, pH can also affect enzymes activity. So you may recall our pH lab, right? The pH scale is an inverse log of the concentration of hydrogen ions in solution. So the pH scale goes from like 0 to 14. Lower the pH, the more acidic it is. Above 7 up to 14 is like your base. So just kind of reviewing that. Well, this can also disrupt the hydrogen bonds of enzyme structure. Basically, we're denaturing its structure. And so just like enzymes have an optimum temperature, they tend to have an optimum pH. But what's interesting about enzymes is it doesn't always have to be neutral, like you might assume. Sometimes their optimum might be in an acidic environment or a basic environment. And let's look at 3 human enzymes here. We're going to look at pepsin, salivary amylase, and trypsin. All of these are digestive enzymes in the human body. We'll start with salivary amylase. What is the pH optimum of salivary amylase? Well, it looks to be just around 7, maybe a little less than that, right? Well, where is salivary amylase used? Well, this is in your mouth. So whenever you eat something and you have saliva, there is an enzyme called amylase in there, and it helps to break down starches. Your mouth has a pH of about, 6.8 to 7.5, right? So it's fairly neutral, which is great for salivary amylase. So salivary amylase will function best at that pH. Now, let's talk about pepsin. Pepsin is a digestive enzyme in your stomach. It's actually released in concert with hydrochloric acid in your stomach. The hydrochloric acid is important to reduce your stomach's pH to like 2. And that's great because pepsin, which is an enzyme that helps to break down proteins, works most efficiently at a pH of 2 or an acidic environment. Trypsin is released in your intestines. And so notice in your intestines, the pH becomes a little bit higher, especially in the colon, where it is more basic, up to about 8.5, which is where the pH optimum for trypsin is. So a lot of the reason why you have stomach acid in different pHs is because your organs are like different chambers for chemical reactions to break down different macromolecules. So we see in your mouth, you are breaking down starches. In your stomach, you're breaking down proteins. In your intestines, you're absorbing sugars and you're absorbing lipids and breaking them down. So each of them is kind of like a little different chemical reaction chamber to break down the food that you eat to maximize the amount of nutrients you get from them. Now let's go ahead and talk about one more thing that affects function, and that is the concentration of the substrate. And so basically, this is what it would look like if we developed a graph. So again, on our x-axis here is the concentration of the substrate, how much of the reactants are available. And the y-axis is the velocity, how much a product is produced per second. And you'll see as we add more substrate to the reaction goes really fast, right? We speed it up, right? So at point A, here we've got 1 microgram of the enzyme available, and the substrate concentration is low, and our reaction speeds up. At point B, notice we have the same amount of enzyme, but we put in more substrate. You'll notice the reaction starts to slow a little bit, and we notice that there is a maximum velocity for the enzyme at this point. At point C, the reaction is slowing down. Same amount of enzyme, more substrate. And then at point D, when the substrate is very high, it's still faster. I shouldn't say this is slowing down. It's not just not increasing. Yeah, I kind of missed it. Let me go back and say this. This is 1/2 of the max velocity is what I should be mentioning here. The reaction at point B is still going faster, but notice as we go from point B to C, the increase in the speed of the reaction is not occurring, it's like not gaining much more momentum, so to speak. We're still making it go faster, but we start to see it level off where there is the maximum velocity for the reaction. So I apologize, I kind of messed that up. But as we add more reactants or substrate, we'll speed up the chemical reaction, but eventually we are going to hit a maximum velocity. Why is this? Well, this visual I think will help with that. When substrate concentration is low, like there's not many reactants, there's plenty of enzyme molecules ready to do their job. So they will make the reaction go real fast. They'll speed it up quicker. When we hit that plateau at maximum velocity, notice we've got more substrate or more reactants than enzyme molecules are ready to do their job. So the enzyme is what we call saturated. There's more substrate than it can work with. And so it can only work so fast. So imagine like it's like workers on a factory floor, right? If you have like 20 workers in a factory putting things together and there's only like 2 orders, man, they are going to put those orders out like crazy, right? No problem. They'll make them quick. If there's 10 orders, well, those 20 factory workers will be able to do those 10 orders, but it might take it'll go fast because they can have 2 workers per person or per order, so it'll go fast, but not as fast as if it was just 2 orders, right? And then eventually, what if there were 50 orders? Well, then you only have 20 factory workers. It's only 20 men. So those orders they'll go out fast because those workers are fast, but there's going to be, it can't go any faster. They just can't work any faster than they're already doing. And that's kind of like what's going on with the enzymes. They just get saturated and they're still doing their job, but they're just not making it go. So just adding more substrate isn't going to necessarily make the reaction go faster. It will to a point, but not once it gets to its maximal velocity or saturation point, it'll level off. One other thing to talk about with enzyme activity is inhibition. So we have 2 types of inhibition that can occur with enzyme activity. And these are important because this is important to regulate your enzyme activity. You can have a competitive inhibitor, which basically will bind irreversibly to the active site of the enzyme. So we call it competitive inhibition because the inhibitor is competing with the substrate for the active site of the enzyme. So notice the blue line here is the reaction of the enzyme with unlimited substrate, right? When you introduce a competitive inhibitor, it'll slow the reaction down. It'll still reach its max velocity, but not as quickly. Inhibition can be important for regulating the speed at which certain reactions are occurring, because some reactions you may not want them to occur too fast. A non-competitive inhibitor will do something similar. It doesn't take more substrate to reach the maximum velocity. It would just not be able to reach the same max velocity. So it basically slows the reaction down like wholesale. And the reason this happens is because the inhibitor will bind to an allosteric site, not the active site. And when that inhibitor binds to the allosteric site on the enzyme, it changes the shape of the enzyme, and now the substrate no longer fits into the enzyme. So that is known as a non-competitive inhibitor. It's going to limit the reaction no matter how much substrate is available. So if you look at the difference between these graphs, competitive inhibitor will delay the reaction's ability to reach its maximum velocity. Eventually, you'll have to add more substrate to outcompete the inhibitor. So you can see it takes more substrate to get to the maximum velocity. Non-competitive inhibition, it's not competing with the substrate. And what the inhibition does is it completely lowers the maximum velocity of the reaction. So it will occur slower no matter how much substrate is there. A cofactor is an ion that will temporarily bind with the surface of an enzyme that can promote a chemical reaction. So it's like a positive, it's like it's the opposite of inhibition. And a coenzyme will also temporarily bind to an enzyme and participates in the reaction as well. So it's kind of like, it's called a coenzyme because it's working with the enzymes. This gets back to the original question I mentioned earlier, vitamins. Why do we take vitamins? Well, let's look at an example of how vitamins work as coenzymes.
This animation demonstrates how B vitamin coenzymes must be present in order to enable some enzymes to carry out their functions. Many of the B vitamins assist in the complex biochemical processes that produce energy from food. The B vitamins are essential parts of coenzymes. Coenzymes are part of the machinery that allows reactions to take place. Enzymes are large protein molecules that catalyze biochemical reactions by lowering activation energies. Enzymes accomplish catalysis by configuring chemical reactants in the best possible spatial arrangement for reactivity. This enzyme catalyzes the release of carbon dioxide when producing energy from carbohydrates. Combining with the coenzyme helps the inactive enzyme achieve the appropriate three-dimensional configuration. For this reaction, thiamine must be present as the coenzyme. The green coenzyme molecule above is thiamine pyrophosphate, the cofactor needed to permit the enzyme and the substrate, pyruvate, to combine in the correct orientation to catalyze the reaction. Now all three molecules are nested together, and the substrate, pyruvate, combines chemically with the coenzyme, thiamine. The biochemical reaction can take place, and energy, as ATP, and CO2 are produced as products of the reaction. Once this happens, the remaining complex, containing the two-carbon fragment, thiamine, and the apoenzyme, rearranges and releases the two-carbon fragment, acetaldehyde, and the ATP. The departure of the acetaldehyde opened the thiamine pyrophosphate to combine with another molecule of pyruvate, and the cycle continues. Meanwhile, the energy is now in the molecule ATP and is available to do work or to be stored. Another way that a coenzyme can influence the reaction between substrate and enzyme is to cause a change in configuration of the enzyme without interacting with the substrate. Here you see the conformational change in the enzyme needed to configure the enzyme to accept the substrate in a lock and key complex. This is called an allosteric control. The cofactor approaches the inactive enzyme and bonds to it in an enzyme-coenzyme complex. The new configuration is a good fit for the substrate. Once this enzyme-cofactor-substrate complex is formed, energy can be released as ATP, and the other reaction products can be released from the complex. Now the correctly configured active site on the enzyme is free to engage another substrate. The amount of B vitamins that are needed to serve as coenzymes is quite small. There is enough thiamine activity in 1.2 milligrams to meet the average person's needs all day. Extra B vitamins will not produce more energy. Excess B vitamins in the body will pass out of the kidneys into the urine. This table shows some details about several of the roles played by B vitamins as coenzymes.